Hello. Good morning, everyone. Good morning, students. I'm happy to be with you today again. Today we are looking at the structure and functions of carbohydrates, the structure and functions of fats, and then the structure and functions of proteins. Inside this topic also, we are going to look at some food tests and photosynthesis. We are also going to look at the organ systems involved in digestion. The structure and function of carbohydrates. Biomolecules are essential for all living things because they are used in a wide variety of vital processes, including the storage of energy and the regulation of metabolic cycles. Some of the most important biomolecules or macromolecules in these processes are sugars, nucleic acids, lipids, and proteins. Every living thing, from bacteria to plants to humans, relies on carbohydrates for basic survival needs. They play a crucial role in our diet, that is carbohydrates, and they can be found in a wide variety of foods, from table sugar and milk to honey and cereals to vegetables like potatoes, yam, cassava, wheat, and many others. Of the four major macromolecules, carbohydrates were the last to receive extensive study. Study these molecules in detail has added to our understanding of biomolecular molecular chemistry by shedding light on how biomolecules change shape and conformation during biochemical reactions. Carbohydrates research has aided our comprehension of fundamental processes such as biosynthetic reactions, enzymatic control mechanisms, and more. In this study, we are going to learn what carbohydrates are, the classification of carbohydrates, and how they are used by various organisms. What then are carbohydrates? Carbohydrates are biomolecules characterized by the presence of multiple hydroxyl groups. Hydroxyl groups means OH group, and a set of naturally occurring carbonic compounds such as aldehydes or ketones. In its most common form, carbohydrates has a carbon to hydrogen to oxygen ratio of 2:1 as in water. The empirical formula for this is CmH2O bracket N. Cm bracket H2O bracket Cm bracket H2O and then N, where M and N are free variables. However, not all compounds adhere to this strict stoichiometric definition. Uronic acids are just one example, and there are others like formaldehyde and acetic acid that share structural similarities with carbohydrates but are not included in this category. Classification of carbohydrates. Carbohydrates are divided into four major groups based on the degree of polymerization, and the groups are monosaccharides, disaccharides, oligosaccharides, and number 4, polysaccharides. We're going to look at each of them one after the other in detail. So without wasting much time, we'll move to the monosaccharides as number 1. Monosaccharides are the simplest carbohydrates and they cannot be hydrolyzed into other smaller carbohydrates. The mono in monosaccharides means one, which shows the presence of only one sugar unit. So in monosaccharides, there's only one sugar unit. They are the building blocks of disaccharides and polysaccharides. So when you have two monosaccharides coming together, it forms disaccharides. When there are more than two, it forms polysaccharides. For this reason, they are also known as simple sugar. So monosaccharides are also known as simple sugar. These simple sugars are colorless. They are crystalline solids that are soluble in water and insoluble in a nonpolar solvent. The general formula representing monosaccharide structure is CN open bracket H2O close bracket N, or CnH2nOn, as shown on the screen. Dihydroxyacetone and D and L glyceraldehydes are the smallest monosaccharides. The monosaccharides containing the aldehyde group, that is the functional group, are the structure RCHO, and they are known as aldolases, and the one containing ketone group is called ketosis. The functional group with the structure RC open bracket double bond O close bracket R with apostrophe close bracket. Some examples of monosaccharides are glucose, fructose, erythrulose, and ribulose. D-glucose is the most common, widely distributed, and abundant carbohydrate. It is commonly known as dextrose, and it's an aldehyde containing 6 carbon atoms called aldohexose. It is present in form in both open chain and cyclic structures. Most monosaccharides names end with the suffix -ose, and based on the number of carbons, which typically ranges from 3 to 7, they may be known as trioses, 3 carbons, tetroses, 4 carbons, pentoses, 5 carbon sugar, hexoses, 6 carbon sugar, and heptoses, 7 carbons. Although glucose, galactose, and fructose all have the chemical formula of CN of C6H12O6, C6H12O6, they differ at the structural and chemical levels because of the different arrangement of the functional groups around their asymmetric carbon. That is what I'm going to show you in the next screen now. You can see glucose, how it appears. 1, 2, 3, 4, 5, 6 carbon atoms. HO attached to the C, to the first C. Then HOH attached to the second. HOH attached to the third. The fourth in glucose down is HOH, HOH, HOH. But in galactose, after HOH, in the 1, 2, 3, 4, in the fourth carbon, we have HOH. And then in fructose, we also have 6 carbon, as you can see here, where the cursor is showing, but the double bond is on the second carbon. These are basically these differences. Even though they have the same molecular weight, they have the same molecular formula, but the structural formula is quite different. So these are the differences we find in these sugars here. Structure of monosaccharide is the next thing we want to look at. Monosaccharides are either present as linear chains or ring-shaped molecules. In a ring form, glucose's hydroxy group can have two different arrangements around the anomeric carbon that becomes asymmetric in the process of ring formation. If the hydroxy group is below carbon number 1 in the sugar, it is said to be in the alpha position. If it is above the plane, it is said to be in the beta position. So we have it here. Conversion between linear and ring forms of glucose is what we are looking at here. So the linear form being converted to the ring form. Then the ring forms of ribose and fructose is shown below in this figure. So here you have glucose up, ribose and fructose down. Look at the structure of both. We can have alpha glucose in ring form. We can have beta glucose in ring form. Then in the case of these ribose and fructose, they are also shown. The issue in the case of glucose is that the carbon that links up with oxygen and then links up back to carbon, in the alpha, the H is in the direction of the O, while in the beta, OH is in the direction of the oxygen. Functions of monosaccharides is the next thing we want to look at. Number 1, glucose is an important source of energy in humans and plants. Plants synthesize glucose using carbon dioxide and water, which in turn is used for their energy requirements. They store the excess glucose as starch, which humans and herbivores consume. The presence of galactose, which is in milk sugar, that's lactose, and fructose in fruits and honey, makes this food sweet. Galactose is also a monosaccharide. Ribose, another monosaccharide, is a structural element of nucleic acids and some coenzymes, while mannose is a constituent of mucoproteins and glycoproteins required for the proper functioning of the body. Now we have looked at the monosaccharides, we have to move on to the disaccharides. 2 sugar units make up disaccharides. 2 sugar, that means 2 monosaccharides, make up disaccharides as a result of a condensation reaction or dehydration synthesis. They yield 2 monosaccharide units. The hydroxy group of 1 monosaccharide forms a covalent bond with the hydrogen of another monosaccharide, releasing a molecule of water. A glycosidic bond is the name for the covalent bond between the 2 sugar molecules. So the 2 monosaccharide sugar molecules that come together to form the glucose, to form the disaccharide, is called glycosidic bond. Glycosidic bonds and links can be either alpha or beta type. When the OH group on carbon 1 of the first glucose is positioned below the ring plane, an alpha bond is made, and when it is positioned above it, it is not alpha, but it is beta. Now we want to see the structure when it is positioned above the ring plane. A beta bond is made. Now we move to look at the structure of sucrose, the diagrammatic representation of the glycosidic bond formation between two sugar units, glucose and fructose, to form a disaccharide, sucrose. So we see it here, glucose is coming together with fructose to form sucrose, and you can see the glycosidic bond being present here between the O and the CO of one and the CO of the other that is being linked together. Disaccharides include sugars like lactose, maltose, and sucrose. One molecule of D-glucose and one molecule of D-fructose make up sucrose, the most common disaccharide, that is sucrose. Technically, sucrose is known as OD-glucopyranosyl 12-D-fructofuranosate, having two sugar molecules, D-galactose and D-glucose. Lactose is found naturally in mammalian milk. Lactose is referred to in the scientific literature by its full name, OD-galactopyranosyl 14-D-glucopyranose. Based on their reactivity to oxidation and reduction, disaccharides can be divided into two classes. Number one class is a reducing sugar. Number two class is a non-reducing sugar. Reducing sugar is a disaccharide in which the reducing sugar has a 3-amiacetal unit, serving as a reducing aldehyde group. Examples include maltose and cellular. Non-reducing sugar are disaccharides that do not have a free amiacetal because they bond through an acetal linkage between their anomeric centers. Examples are sucrose and trehalose. Disaccharides include but are not limited to xylobiose, manobiose, melibiose, rutinose, melibiulose, palotinose, trehalose, dentiobiulose, maltulose, chitobiose, and many others as shown here. What then are the functions of disaccharides? Number one is that sucrose produced during photosynthesis provides plants with both carbon and energy. Number two is that lactose is an excellent source of energy for animals. Number three is that maltose plays a crucial role in the breakdown of starch and glycogen. While number four is that trehalose is a vital source of energy for insects. Number five, cellulobiose is a component of all glycosides and some polysaccharides found in plants. Dentiobiose is a component of plant glycosides. We'll move on to the third carbohydrate, which is oligosaccharides. When hydrolyzed, oligosaccharides can release anywhere from 3 to 10 monosaccharides molecules. Glycosidic linkage connects all the monosaccharides together. Additionally, oligosaccharides are categorized as trisaccharides, tetrasaccharides, pentasaccharides, etc., based on the number of attached monosaccharides. Trisaccharides have a general formula of CN open bracket H2O close bracket N minus 2. Tetrasaccharides has the same thing, but it's N minus 3, and so on. Glycan forms of the oligosaccharides are the most common form. These molecules form as glycolipids or glycoproteins and are joined to lipids or the amino acid side chains of proteins via N or O glycosidic bonds. Glycosylation is the process by which a carbohydrate is covalently attached. Take note of this terminology. Glycosylation is the process by which a carbohydrate is covalently attached to an organic molecule, producing structures like glycoproteins and glycolipids, which are held together by glycosidic bonds. Glycosidic bonds. By forming a beta linkage to the amine nitrogen of the side chain, oligosaccharides can be attached to asparagine to form N-linked oligosaccharides. A membrane of the endoplasmic reticulum is the site of this procedure in the eukaryotic cells. In contrast, in prokaryotes, this process takes place at the cell membrane. Attaching oligosaccharides to the hydroxyl group of a side chain amino acid like threonine or serine is what is known as O-linked oligosaccharides. O-linked oligosaccharides. It is a process that takes place in the Golgi apparatus. It is a process that takes place in the Golgi apparatus, where monosaccharide units are added to an already complete polypeptide chain. What then are the functions of oligosaccharides? Number one, they have critical functions such as antigenicity, solubility, and resistance to proteases are performed by glycoproteins, which are carbohydrates attached to proteins. At receptors, adhesion molecules, immunoglobulins, and tumor antigens on cell surfaces, glycoproteins play important roles. Cell recognition and regulation of receptor membrane proteins are facilitated by glycolipids, which are carbohydrate attached lipids. Cells produce lectins, which are carbohydrate binding proteins that facilitate cell division, cell adhesion via oligosaccharides. Oligosaccharides are a type of sugar that are found in plant fiber. We now move to the next one, which is number 4, polysaccharides. As the name implies, poly means multiple, many, plenty. More than 10 individual carbohydrates are linked together in a chain by our friend the glycosidic bonds to form a polysaccharide. They are found pretty much everywhere and serve mostly structural or archival purposes in living things, also called glycans. It is the composition and structure of the binding or reacting molecules, as well as their interaction with the enzyme machinery, that determine the physical and biological properties of these compounds. Polysaccharides can be broken down into subgroups based on their roles, the types of monosaccharide units they contain, or their biological origin. Homopolysaccharides and heteropolysaccharides are two categories of polysaccharides, distinguished by the types of monosaccharides used in their formation. Homopolysaccharides, they have only one kind of monomer unit repeating in them: xylan, chitin, glycogen, and starches. Amylose and amylopectin are all examples of homopolysaccharides. There are two main types of polysaccharides: structural polysaccharides and storage polysaccharides, both of which have different purposes. Glucose units are linked together in cellulose via beta-1,4 linkages, making it a linear unbranched polymer, one of the most prevalent organic compounds in the biosphere. Chitin is made up of 1,4 glycosidic linkages between N-acetyl D-glucosamine, a glucose derivative, residues along a long linear chain. After cellulose is the most common biopolymer found in nature. Alpha linkages connect the repeating units of D-glucose that make up starch. Alpha linkages, I'm taking that again because it's important. Alpha linkages connect the repeating units of D-glucose that make up starch. A mixture of 15 to 20% amylose and 80 to 85% amylopectin. It is one of the most common polysaccharides in plants. So we are now looking at the structural representation of cellulose. So we have the structural structure of cellulose here, and we can see it being shown to us. Now, the structure is longer than this. As I said, it's about 10 monosaccharides, more than 10 polysaccharides or more. So the end that is showing here after this bracket is showing that it can go more and more, out to 20, 30, 40, 50, 60. So this can keep on repeating until it gets beyond 10 or gets to 10. Heteropolysaccharides, they have two or more types of monomer units that repeat. Glycosaminoglycans, agarose, and peptidoglycans are all good examples. They are connected to proteins, lipids, and peptides in natural systems. Negatively charged unbranched heteropolysaccharides are glucosaminoglycans, repeating units of disaccharides with the general structural formula and make up their entirety. The glucosaminoglycans GAG structure typically includes amino acids like N acids, like glutamic acids. Peptidoglycan is a beta-1,4 glycosidic linked heteropolymer of N-acetylglucosamine and N-acetylmuramic acid. Agarose is a polysaccharide made up of chains of disaccharide agarobiols, which consists of the galactose and 3,6-anhydro L-galactopyranose. 'L' galactopyranose. 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 'L' galactop So here we have the structure, classification summary, polysaccharides into different subgroups, from polysaccharides to homopolysaccharides, heteropolysaccharides, storage polysaccharides, and many others. Now let's look at the functions of polysaccharides before we move on to the next aspect of the topic. Structural polysaccharides, for example, give tissues, organs, and organisms a solid foundation from which to function. Chitin and cellulose are a couple of examples. Cellulose is a critical part of the diet for ruminants, and chitin plays a role in the synthesis of fungal cell walls. So we can go on and look at the benefits. So here we have classification summary. An example of carbohydrates is shown in this table, and then a final words or as a conclusion. Carbohydrates are a type of sugar that is needed by all cells in the human body. The number of monomer units in their structure determines which of four groups they belong to and the forms of which they are consumed by organisms. Monosaccharides, disaccharides, oligosaccharides, and polysaccharides are all types of simple sugars. Glucose, fructose, cellulose, starch, glycoproteins, and chitin are just some of the molecules found in carbohydrates. They serve as a wide variety of purposes, from fueling the cells to ensuring the cell's structural integrity is maintained and to promoting development and growth. Studying carbohydrates has helped scientists understand the role of conformational changes in molecular kinetics and other processes, and scientists who study these molecules still have a lot to learn about the many sets. So we're going to continue the next class after this break.